Hi, my name is Kyle Fink. I'm an assistant professor here at the UC Davis School of Medicine, and my lab is in our stem cell program, gene therapy center, and part of our MIND Institute. And I'm here today speaking on behalf of our wonderful colleagues as part of our interventional genetics group and interventional genetics team, consisting of myself, Dave Siegel, Jill Silverman, and Julian Halmai, who is an outstanding postdoc currently in my lab. And what we really wanted to talk a little bit about today is, the scientists, so you can have a face, you can meet us. My lab really focuses on gene therapy. Jill is a world-renowned expert in animal behavior and testing translational phenotypes in mice and rats. And Dave Siegel is really one of the premier pioneers in the world of genetic engineering and genome modification. And we're very excited to be speaking on behalf of the ACNRF as well as CRF, the CASK foundations that are very focused on developing novel therapeutics. And what we wanted to talk a little bit about today is a new project that we're very hopeful to launch within our lab. And really, for just a little bit of background on what we're doing is my lab is very interested in X chromosome biology. And how the X chromosome inactivation plays a role in some of these X-linked intellectual disability genes. And we've been working on several of these different candidate genes for several years in my lab, really led by Dr. Julian Halmai. And how we can think about X chromosome inactivation is all females have 2 copies of the X chromosome, and we can see in each individual cell represented by the circles in the brain here is it doesn't necessarily matter which one of these X chromosomes is active. And so we typically refer to this as the active X chromosome or the inactive X chromosome, as you can see delineated here between gray or dark gray and light gray. And so in a normal situation without a CASK variant, as you can see that both of these CASK genes are healthy and intact, whether or not it's on this chromosome on the left or the chromosome on the right in these 2 different cells. And what that will look like is that every cell in the brain will be expressing a normal, healthy copy of CASK. What we see in CASK-related intellectual disabilities is if there is a variant on this right chromosome, you can see here, this right X chromosome, is that in the cells that have the left chromosome active, as you can see here in blue, is these neurons are expressing a healthy version of CASK. In the cells in which the active X chromosome is lacking or missing this CASK gene, you can see these are white. And what this means is that through random X chromosome activation and what we refer to as a mosaic brain, is approximately 50% of those cells won't be expressing the needed CASK protein. And that's what really results in a lot of these neurological problems in the patients with these CASK variants. And so what one of the goals of our lab is to do is to be able to go back in, because on this left chromosome you can see in the middle, they have a healthy and intact CASK protein that we're going to try to turn back on or reactivate. And we call this targeted X reactivation, artificial escape. And really what the ultimate goal would be is in these 4 neurons, or 50% of the neurons in a brain, is that we can induce these and have them come back on and be expressed. And we do this through some really cool technology that a lot of us in the scientific world are working on now, using things like CRISPR Cas9, not to cut the DNA, but to regulate and modify the gene. And so normally what we think of on that inactive X chromosome, what we're referred to as XI, is that the inactive gene is heavily methylated and that gene is turned off. And so by looking at different types of what we call effector domains, or these epigenetic editing domains, you can see as indicated by these different colors, is we can go in and try to force the expression or turn that gene back on. And we've been successfully doing this in other X-linked intellectual disabilities like Rett syndrome and CBK5 deficiency disorder, where again, we can land these different constructs to open up this healthy copy of the gene, producing mRNA and hopefully protein to restore function. And what our lab really does is comprehensively study this in terms of developing the therapeutics and testing that in cells to be able to move it forward, checking what's happening in terms of gene expression, if we can actually get reactivation, looking at what happens in patient-derived iPSC, induced pluripotent stem cells that we can turn into neurons and measure what that function looks like. And then eventually testing it in transgenic animal models. So you can see kind of in A, this would be where we have the gene off. That variant or mutant allele is on the active X chromosome. Our patient cells, our patient neurons, or these diseases in a dish look unhealthy, and that we can see phenotypes across the animal models of the disorder that we use for testing. And so when we come in and we actually bring our Cas9 construct, ideally we can turn that gene back on, we can see it reactivate at a molecular level, we can see functional restoration of cells and ideally rescuing the phenotypes. And this is really what we're planning on doing, that we're very excited to launch and to partner with the Patient Foundation's research program targeted at developing this type of therapeutic to hopefully make an impact in the lives of those affected by these CASK variants. And again, it's great to be a representative of our very comprehensive and large group here at UC Davis that is very focused on developing these types of genetic interventions for rare disease. Thank you.